Let's get right to it. We're going to be covering some basics on genetics today. Nothing in much detail. I do apologize ahead of time because this period is really giving me some pretty bad allergies. So my voice, or I might cough or feel slightly short of breath. So I do apologize as I go through this. I'll do my best. So do you guys remember, or how many of you remember back, in biology doing genetics? You remember studying Mendel and the peas, and what's the difference between a dominant or a recessive allele, or what is an allele, for example, right? And so the area of inheritance really has intrigued a lot of us, even probably before even Mendel, although Mendel from a scientific perspective was probably one of the first to manifest it or bring it forth. And he made his observations, with bean plants and whatnot. And then we moved forward to the discovery of the double helix and James Watson and Francis, what was his name? Crick, I think, discovered the DNA and as we move forward, remember as registered nurses, healthcare providers, we don't need to know everything. And in fact, with all the amount of information out there, it's impossible to know everything. But it's not a bad idea for all of us, as we go through, as we enter into our career as nurses, start thinking about the availability and what tools and what science is starting to bring forth. And some of you may want to go into this field of genetics, epigenetics, genomics, nutrigenomics, you name it. And so at the end, I'll give a little bit of a brief introduction to other terminologies that will introduce you to sort of where we're at today and what's going on. So without further ado, let's get into it. There are some objectives. The genetic terms. There's basically 2 or 3 inheritance patterns. That's what you will be expected to know. We'll look at some basic genetic mutations, most common classifications of some of the genetic disorders, and then we'll look at not only pharmacogenetics. I added also nutrigenomics, because as we move forward, we are now starting to discover things in plants and foods that can actually target alleles or target the DNA without really making change to the DNA. And then we'll sort of summarize some of the social implications with genetic testing, who should give it, who shouldn't, and we'll describe what our role as an RN is in helping the patient or the family in dealing with genetic issues. Now, if you're working in the typical acute care setting, ER, ICU, step-down progressive care unit, telemetry, medical, surgical, or surgical, you won't be dealing with a lot of these issues that we'll cover here, but it's still not a bad idea to get familiar with the terminology and understand it in its basics. And if you're very interested, then maybe even move into it as an area of specialty. I found this quote that I found very interesting, and I wanted to share with you guys by the director of the United States National Human Genome Research Institute. His name was Francis Collin. He was appointed and sworn in as the 16th director of the National Institutes of Health in 2009, nominated by President Obama. And so he has a really interesting comment that I want to read to you that was used as evidence to the United States House of Representatives Committee back in 2006. And I think it gives us a good foundation for which to build upon this talk or lecture about genetics. So let me read this for you. You can read along. Recent increases in chronic diseases like diabetes or childhood asthma, obesity, or autism cannot be due to major shifts in the human gene pool, as those changes take much more time to occur. They must be due to changes in the environment, including diet and physical activity, which may produce disease in genetically predisposed persons. Therefore, GEI, the institute that he's working for, the research institute, will also invest in innovative new technologies and sensors. By that, I believe he's talking about biomarkers to measure environmental toxins, dietary intake, physical activity, and using new tools of genomics, proteomics, and understanding metabolism rates to determine an individual's biological response to those influences. And I found this very interesting that the NIH, the Genome Research Institute, is saying, yeah, we're going to be looking at the genetics, but what we want to also look at, not completely ignore the genetics, but what we want to look at is the interaction that individual genes in different people have with the environment that we're living in, the food that we are consuming, the air that we're breathing, the jobs that we may have that might put us at certain toxic risks, and so on and so forth. Another thing that I found from the American Medical Association was Garth Graham, a medical doctor who is the president of one of the Aetna Foundation, I think insurance companies here. And one thing that he said during a presentation is, that for a lot of people, personalized medicine really is about targeting treatments based on the genetic profile. But as we move more upstream, we're starting to realize that, and many physicians have started to realize that really personalized medicine, yes, it means looking at that genetic profile, but also has a different meaning. And I'll open quote, a person's zip code, not the genetic code, but a person's zip code matters more than their genetic code, end of quote, right? During a presentation he did, open quote, where people live really defines their health outcomes. And so that's what I wanted to bring forth to the table, is that you take genetics with a grain of salt, because you may have the genes for disease, and if you do the right things, you may never manifest a disease. You may not have the genes for disease, and if you do the wrong things, you may manifest a disease. So genetics, depending on who you ask, some people say as low as 3 to 5%, other people say 5 to 7, some people say up to 11, 12, maybe even 15%. But even if it were 15%, that's a large percentage of environmental factors that are going to be turning on the genes to lead to disease or health. So keep that in mind as we move through this lecture. So let's review some terms, right? Remember what you have a nucleus, and inside the nucleus you have chromosomes, and the genes are part of that chromosomes, right? And so when I ask you what contains the genetic blueprint of you, me, or your mom, your dad, your children, it's the chromosomes that contain the genetic blueprint, right? And where are these chromosomes? They're contained in the nucleus of every single cell, right? And so remember, chromosomes are what contain the genetic blueprint, and they're made up out of what? Well, they're made up out of DNA, deoxyribonucleic acid. And what's DNA made out of? Well, quite a few things. Nitrogenous bases that really ultimately maintain life, right? And genes are a specific part of the DNA that contains inheritance, the specific hereditary information that we pass on to our children, or that our parents pass on to us. Also, what's contained in these genes is how control of how a cell works and functions, right? It helps produce proteins to perform hundreds of thousands of tasks, and they can act as cell messengers. They can, the role is incredible. Now, RNA is a little different because that's more of the transferring of the genetic information. Remember that from like DNA for protein synthesis. And then one thing to remember with the chromosomes is mitosis and meiosis. Anybody remember the definition of those? Mitosis being the cell is dividing, which results in identical daughter cells. That's mitosis. And meiosis is the cell that's dividing or happens through sexual reproduction. So it results in half of the number of the chromosomes. So it's quite different from mitosis. Well, God, what were some of the other things? Well, there's autosome, which is pretty much chromosomes that's not sex chromosome. And how many pairs do we have as humans? 22 pairs. then there's going to be mutations. When we talk a lot about mutations, we're talking about also this nucleus chromosome genetic information is what's being mutated. It's the change in the normal sequence of DNA, which could lead to either disease or putting the patient at a risk for disease. And then two words I do remember, two words that we want to really learn very carefully, and we'll talk about them as we go through, but I'm mentioning them as a heads up. Heterozygous and homozygous. And heterozygous has two different alleles for one given gene, remember, one from each parent, and homozygous meaning having two identical alleles for any one given gene, also one inherited from each parent, sorry. So let's get into a couple of these. Remember, phenotype and genotype are important terms to learn and remember, because phenotype, think of it this way. Here you have a baby, right? And the infant has blue eyes, right? So what is the phenotype of this baby and what is the genotype of that baby you're looking at? Well, think of phenotype as what you can see, and what's being expressed, is what's obvious: brown hair, blue eyes. So in this infant, the blue eyes are the phenotype. The genotype is the actual genetic sequencing, which we can't see with our physical eyes, that causes the blue eyes to be expressed. So underlying every phenotype, there's a genotype. Okay, that's important to remember. And then alleles are you look at the chromosome and you have different types of phenotype, and you may have an allele for phenotype A and an allele for phenotype B, and they're ultimately what will express in the, as I said previously, eye color or hair color. The alternative forms of a gene that's brought down to us. This is part of inheritance that we are going to manifest. And that's what phenotypes mean. So remember that. And alleles, we'll talk about that shortly. So patterns of inheritance or mutation, or how things happen, right? So let's think of, in this case, autosomal dominance. And you need to remember what autosomal dominance is. And males and females will be affected equally. And they are more common and usually not as severe as recessive disorders. So recessive disorders, remember, are going to be, or can be, more severe than autosomal dominance. And also autosomal dominance, they have variable expression, meaning they may have an affected adult, for example, or one way, I guess, of putting it is a dominant trait is always going to be the one that's expressed, right? And if the allele, remember from the previous slide, is present, it's going to be expressed. So I'll go back to this. So if you have phenotype A in these alleles, or phenotype B, whichever of these two is more dominant, right, that is going to be the one that will manifest itself. So if the allele is present and there's more than one different alleles, the one that is autosomal dominant will be the one that expresses itself. For instance, brown eyes or brown hair. Now, some diseases also are dominant, right? And some examples are breast cancer or ovarian cancer, and I believe that's related to the BRCA gene or genes. Polycystic kidney disease has some inheritance, some dominant. Family high cholesterol is also an autosomal dominant. So those are good things to keep in mind. Now, continuing this inheritance or mutation problem, let's look a little bit more at autosomal recessive, right? And males and females will equally be affected. Now, in this case, individuals with a heterozygous trait are carriers. They may not have any symptoms, but the affected individuals may have unaffected parents who are heterozygous for the trait. So let's think about inheritance. So if you have children of two heterozygous parents, then they'll have 25% chance of being affected and 50% chance of being a character. And there may be no family history or trait of that disease. And then, this, when we talk about the 25 or the 50%, this is, as you've had in some of your prereqs to get into the nursing program, creating that Punnett square to try to draw that, which we're not going to do in this class. Now, for example, there's some groundbreaking research on trace minerals, for example, at, I think it was Emory University back in the 70s. For example, Dr. Wallach discovered what was seen as the first known case of non-human cystic fibrosis. And he declared that it was, by and large, a selenium deficiency. He found it in monkeys. And at the time back then, cystic fibrosis was believed to be the result of a human genetic disorder. But as we discover and science starts to come more to the forefront, we start to realize that that sometimes having a deficiency, perhaps as selenium or a trace mineral or a micronutrient, may lead to what seems to be a mutation. But in reality, the DNA, the essence of the DNA, may not have changed. As long as we supplement proper selenium, then a lot of the traits may disappear, and this may start to happen as we move through. Even I believe muscular dystrophy as well, as they found out in livestock industry, and they found that some of these livestock were just eating foods from soils that were depleted of minerals, and that some similar symptoms that manifested similar or as muscular dystrophy disappeared with just simply filling the soil back up or giving them the supplements. So we have to take the inheritance with a little bit of a grain of salt. But generally speaking, cystic fibrosis is seen as a classic autosomal recessive pattern. Now, the X-linked recessive, who do you think this one affects more? Equally male and female? Or usually males? Well, the word X-linked recessive should tell you that it's mostly males, because females are going to have another chromosome that will override the recessive trait. And so it's mostly males. Now, daughters of affected males can be carriers, and sons may be unaffected unless the mother also was a carrier. And the two examples here are hemophilia and the Duchenne muscular dystrophy as well. Now, there are some X-linked dominant. Don't worry about that; those are very rare. But do know that the X-linked recessive are usually in males. This is an example of the hemophilia. I'm not going to go into this pattern. It's an inherited disorder where the blood doesn't clot because of a lack of clotting factors, basically. And we always go over the Queen Victoria and Prince Albert had 9 children, and how 5, I think 4 or 5 didn't receive the trait, and 4 did, and expected 3 girls were the carriers, and their son, Leopold, ultimately was the one that was affected. This is a classic genetic discussion that one has. Now, there are diseases that are a result of, and many of the chronic diseases that we're seeing today are thought to be multifactorial inherited conditions. But we'll talk about a word that I think is becoming perhaps slightly more important than genetics, and it's epigenetics, or above the genes, above the genetics. And that just means that food, environment, toxic air, pollution, mercury, asbestos, glyphosate, which is in Roundup Ready, all of these things can end up going into your body and leading to what seem like they're really genetic conditions. They may not necessarily have fully or completely changed the DNA, but definitely manifest a lot of diseases. And so epigenetic, such as Roundup Ready and mercury and smoking cigarettes and eating processed foods, high fructose corn syrup, all of these can ultimately affect the body in many different ways, leading to chronic conditions like diabetes, obesity, hypertension, cancer, coronary artery disease. The interesting thing is that there's a lot of people who have no diabetes mellitus in their family, and they get it, especially type 2, right? That's lifestyle related. Obesity. There may be, I remember we used to be looking for the obesity gene. It's a lot more complex than that, right? The adipose tissue, they have a brain of their own. They can block satiety center, hypertension, cancer. So a lot of people end up having these chronic conditions and don't have family history. And a lot of people have family history, don't necessarily have to end up with the condition. So as nurses, we need to be aware of that because a lot of times I think that the risk is to communicate with our patients that, oh, you have a family history of high cholesterol or family history of hypertension or family history of cancer. There's really not much you can do. By saying that to your patients, you're really putting them in a spot where they're just going to probably just eat even worse. After all, there's nothing I can do. Why even try? Not true, not true, not true, people. Epigenetics is starting to demonstrate without a shadow of a doubt that all of these conditions can be reversed. We know with hypertension alone, eating a DASH diet, right, sort of close to that Mediterranean diet, this is a very good thing to know. We know that CoQ10, magnesium, can help boost the body to work with hypertension as well. Coronary artery disease, right? Same thing. Diabetes, same thing. So there's a lot of, we need to give power to our patients, not take it away. So in these multifactorial inherited conditions, all of the chronic conditions that are killing most of us in our world and here in the United States, there's a lot of power that we have in our hands. And so don't be going around saying, oh, because it's genetic or it's in your family history, that's it, right? No, that's not true. You could have it. We need to be saying is you could have it in your family, and doing the right things in your life and lifestyle never manifest it. And others who don't have it as a family history, doing the wrong things, could clearly manifest it. If not, ask your patients that you meet who have hypertension or diabetes or obesity or coronary artery disease. Clearly lifestyle related, which can trigger the genetics and changes, right? That's why we call it epigenetics. Now, there are common causes of genetic mutations. The germline mutation usually passed down from a parent to a child. And here are, again, examples that were repeated, like the autosomal dominant on the left, autosomal recessive, so these are present in the sperm cell, right? What was the oocyte, and the mutation is present in every cell and for the individual's lifetime. And you'll see which one of the will end up carrying it. Affected on the left are the orange ones, 2 out of 4 with autosomal dominant. The autosomal recessive ends up having child with the mutations. Only 1 out of 4. 1 is the carrier. 2 are carriers, and 1 has no mutations. And then the other one is the sample of hemophilia that we talked about earlier. So common causes of genetic mutations, again, are, remember, think about acquired. Somatic mutation are going to happen within the DNA of a cell, right? And they're going to be acquired mutation. And these are passed on to all the cells that develop from that cell. They will not be, these acquired, because they're acquired, remember this, they're acquired mutations, they cannot be passed on to the next generation. This is not part of, when you hear acquired mutation, we're not talking about inheritance or passing things on to the next generation. Usually happens, again, same thing, cell division or caused by environmental factors like UV light and skin cancer, or toxins like asbestos, right? Or hairy cell leukemia, or toxins in cigarettes leading to lung cancer. Chemotherapy, which increases the incidence of secondary cancer, for example, right? These are, the chemotherapy is trying to help, but at the same time, if it produces an acquired mutation in other cells, all of those cells will reproduce it from then onward. But if that person were to have children, that will not affect them. Also, acquired mutations or somatic mutations can occur because there's a mistake during the DNA replication, a deletion or insertion, or when they duplicate themselves, they don't do it properly, or there's an error. and acquired are generally very much related to environmental factors that will affect the DNA and produce ultimately disease. Single gene disorders are, as it says, a mutation of a single gene. Cystic fibrosis, sickle cell, polycystic kidney disease, or PKD. Again, these may be autosomal dominant, autosomal recessive. They could be even X-linked. They're relatively rare compared to the multifactorial genetic disorders. So the multifactorial are significantly more common. Small changes in several or many genes can act together. Again, if there's atherosclerosis or cholesterol buildup in your family, yeah, you may have some genetics, but remember, genetics is 2, 3, 5, 10, 11, 12, 15%, depending on what literature you read. The other percentage is what you're doing with your own life and what's leading to that. And remember, we've talked significantly a lot about coronary artery disease and the buildup of the plaque, and a lot of it has to do with the lining, the endothelial lining. It's weakening, which leads to micro ruptures, which lead to the plaque buildup. So the plaque is not the problem; it's what's underlying the plaque that leads to it. And so, proper lifestyle and diet, especially, can make a big difference. Now, yes, genetic mutations will predispose the individual for developing a disease, and the environmental factors can alter the gene expression. They can switch things on or off, right? That's what we call epigenetics, right? The studies of how gene functions are being changed without ultimately changing the DNA sequencing. And that's what's happening in a lot of our chronic disease, chronic problems that we're seeing today. Now, trisomy 21 or Down syndrome is a chromosome disorder that happens when meiosis, that the sperm ends up retaining both copies when that fertilization occurs, and the embryo ends up having the 3 number 21 chromosomes instead of 2. And it's one of the most common genetic defects. There are other types of Down syndrome that affect the chromosome 21 as well. I think translocation of chromosomes is one, and also the mosaic Down syndrome, which could be which is the mosaic Down syndrome, which you don't need to know is it happens after the fertilization happens. The most common one is the one that happens during fertilization, which is trisomy 21. That happens during the meiosis process. We'll get into this a little bit more down the line. And yes, pharmacotherapeutics are really looking into genetics and epigenetics and how certain cytochromes and alleles that people carry then will let us know if you need an antihypertensive medication, but you have a very specific allele, then don't give this med because it's not going to function as well as if you give this other one. And so that's the notion of looking at personalized drug therapy for the patients. But as we learn all of these things, we also learn that food as well has a very specific metabolism and alleles that can affect patients as well. I guess in the hospital, one classic example is fast metabolizers or slow metabolizers that may affect drugs like Coumadin or Plavix, right? And how that affects, if they metabolize it faster or slower, then how much drug they get or which drug they get, or are they more at risk for bleeding, is really important to know. And there's we'll talk about this shortly. You don't need to know these, but again, in pharmacogenetics, when we do develop personalized drug therapy, there are certain gene mutations that people have, like the BRAF V600E or the ALK or the HER2 protein. And so this is sort of the new gene therapy. It's the, that we're targeting patient treatments based on the gene mutations that they have or don't have. And so the new gene therapy would be injecting genetic material into a cell to cure a disease, right? And this is really still in the experimentation stage. So we're far yet from it, although with CRISPR technology, a lot of wonderful things are starting to happen. But in research, there is showing some promise. So we'll just keep your eyes open and be open to what we discover as you go through the next few decades of your career as a nurse. Stem cell therapy is basically taking a stem cell, which means a cell that is not specialized, and that can end up developing into a specialized cell. So if you have a unspecialized cell or a stem cell, and you can create it, may allow it to turn into a muscle cell, a blood cell, a nerve cell, a cardiac cell, a liver cell, an intestinal cell, right? And the reason for this is because we can use them to repair or replace tissue. If there's been tissue damage, we can replace it. Now, embryonic stem cells, as the word says, comes from the embryo, and they'll develop into any one of the many different cell types. They can be stimulated into pretty much any type of tissue. Similar to the stem cell, the embryonic can get in. And where do we find these? Usually in the umbilical cord blood, for example. So stem cell therapy is also showing some very, very promising treatments as we move forward as well. Now, there's two different types of genetic testing. There's medical or predictive. And God, there's thousands of diseases that we can now look at to determine if the patient has certain results in their genetic testing. We can use them to diagnose the disorder, including cystic fibrosis as well. Down syndrome, as we talked about, the Duchenne muscular dystrophy, for example, is a genetic testing it's more of a medical than a predictive. It's done to diagnose more than anything. Now, a lot of the insurance companies, unfortunately, still won't cover the cost of a lot of this genetic testing. Yeah, so let's move into the next slide. So ethical and social implications of genetic testing. So who should have access to the results, right? I mean, let's say they do some research on you and find like there's some cells or genes that they could use to culture and create and utilize for certain therapies. Should you be informed, or do the healthcare professionals have the right? I think it was Henrietta Lacks, African-American female, had cervical cancer, and she had a tumor removed, I think in Johns Hopkins, and these cells were then cultured, and they found that they replicated, and these cells became known as the HeLa, and they're still used for medical research. Did she probably get any benefit from that? I mean, after all, it was her body, it was her own. So these are issues that we need to be transparent and clear about. They're complicated issues. Genetic testing can reveal information that can affect family members, or when a whole family is tested, it could reveal relationships that are different than what were thought, right? And there was an act back in 2008, Genetic Information Non-Discrimination Act, that protects people from discrimination by employers or health insurance companies because they found something on the genetic. Otherwise, if that hadn't been put in place, then nobody would want to get tested, and for fear of retaliation, right? Because you have to think that insurance companies used to be able to refuse coverage for pre-existing condition. Think about that, right? Now, when parents are seeking treatment for their child, right, they could be asked to participate in studies, and who has the right to that study information is also a big problem, right? And then um, there's a lot of examples. So who's protecting the privacy? Is it the hospital? Is it federal? Is it state? Is it county? Is it so just make sure that your patients, if they ever do end up getting involved, or you're working within the field of genetic testing, that you inform your patients fully to know what their rights are and what rights we will be keeping in the hospital or in the outpatient setting to move research further, and they will need to sign or not. Whether they agree or disagree. Remember, it's just like an informed consent. Somebody's going into a surgery, an informed consent means they're consenting because they've been informed. So always, always, always inform your patient. And ultimately, what's the bottom line of this slide? I would say subjects, I hate the word subjects, individuals, people, humans, ultimately have the right to determine who has access to the records for their study, right? No different from the HIPAA as well, so we have to be careful. So the role, as you've already done your genetic paper, by the way, quite a bit, very, very, very well done. I'm surprised a lot of you did a really good job. And what basically you're doing is looking at your own family history, right? And because we know that family history really has strong influence on risk, right? If there's two people, one has family history of high cholesterol or hypertension and one doesn't, one is going to be more likely prone to getting hypertension or high cholesterol. Again, epigenetics plays a major, major, major role in it. And so we need to, for chronic disease, keep that in mind. But the role that we will be playing as RNs, if you do go into this field, mostly in the hospital, on the floors, you won't be utilizing this, but you do now know how to create sort of a blueprint portrait of family members and who has what diseases to create a family health history, a genogram, right, is what we're talking about here. And this activity will help see patterns of disease within a family. And patterns of disease is important if they can go back. Sometimes when you get patients to do that, they didn't know that their dad died of something specific or that their grandparents had a specific disease. And being informed is good, because if you now know, there may be literature coming out saying that eliminating certain things from your diet or adding certain things to your diet could help the problem that your grandfather had or grandmother, or may even prevent it, or may even make it worse. So knowing and information is clearly having power. And so we can work with the patient to create a genogram. We can talk about the risks that they're going to be at, and then we can talk to them about what changes they may need to be doing in their life, like cholesterol, eat less saturated fats, eat more of the better fats, and so on and so forth. Stop smoking, things of that nature. So our role is ultimately to promote health, right? We should encourage screening and testing, and we should promote lifestyle. And screening, really, we're talking about more of the secondary, right? The colonoscopy, lipid panels, mammogram, primary, secondary checking. But then lifestyle, again, we know that lifestyle can completely help a patient in their chronic diseases get through it. And we know that eating processed foods is clearly has negative effects on the patients. And so we want to always talk to our patients to, for one, just stop eating any of the processed foods, eat as much natural food sources as possible. And we know that getting out and exercising, breathing fresh air, de-stressing, going out into nature, listening to music, having a pet, sitting down and eating a meal with your family, living in a neighborhood where you have more than just stores that sell alcohol. It's making sure you spend time with your family and friends. and definitely not smoking, right? We know that adopting these positive lifestyles have an effect on genetic manifestation of diseases, no doubt about it. And so we can support families by helping them. Some families may decide to have genetic testing done, right? And they'll need to get some genetic counseling. And this is where, if you're an RN who works in this field, you'll be working in a lot of the genetic counseling, and you'll need to learn that people can do home genetic testing at home. They can come in and do it in an outpatient setting in a clinic. But a lot of times, if they're doing it at home, they may not always have the access to counseling unless there's a phone number, or they have a follow-up with these people to sort of interpret the results, and that won't be helpful for anybody. So if there are kits that are going home, if that same company provides counseling, it may very well be a registered nurse who will call the patient up or meet the patient face to face to discuss what's going on with their inheritance patterns and reinforce accurate information and advocate for the patient improve, right? They need somebody who's knowledgeable in the field to interpret a lot of the results, is I guess what I'm saying, and to guide them in the directions that that'll be beneficial for them to move forward. As our 21st century medicine really comes forward, we're moving away from an acute care model to really a far more effective sort of chronic disease model, whichever model that may be. But that will only happen if we start attacking the underlying drivers of the problems that we're seeing, right? Which is really complicated reactions between the lifestyle, the environment, and the genetics. So it's not the genetics alone, it's not the environment alone, it's not the lifestyle alone. One has to, yes, look at the lifestyle, but look at the environment that one lives in as well. And if we do engage our healthcare system in an effort to implement these changes, it's going to be beneficial for everybody, because the big killers, myocardial infarctions, strokes, heart attacks, diabetes type 2, are clearly related to, yes, there may be some genetic vulnerability that may play a small percentage of it, but ultimately the environment, toxicity, the homogenization and denaturing of the food supply, which is why we say if you can buy local, go to the farm, support your farm. Homogenization is where you buy food in big, large stores that are selling that produce foods like we would produce robots in the big chain, sort of, right? They use pesticides or they give hormones or antibiotics to the animal, and if the soil is overused, it's depleted of the nutrients. So the food supply, if it's being denatured, obviously that's going to play a major role, right? The influence of sedentary technology, whether you sit down in front of a computer the whole time, whether you're educated or you're constantly being exposed to entertainment at home. And also there's the issue of the powerlessness and despair of poverty, right? Debility can lead to chronic stress, and then there's fragmentation of family and fragmentation of communities. And all of this leads to an isolation and a lessened sense of purpose and meaning. So the danger with going down this track of thinking about everything is about nutrition, everything is about lifestyle, and eat the broccoli and the lycopene and so forth, the lycopene in the watermelon or the tomato, it can help you for X, Y, or Z. That's a very important aspect that we need to talk to our patients with. But we also need to add into the discussion that community life is important, and sometimes talking about trying to bring back to their being a sense of purpose and meaning is as important or more important than genetics. So keep that in mind. Again, prediction is when you have technologies or tools where healthcare providers can utilize. They examine genetic makeup. The physician then can look at, they can make some comprehensive predictions about the patient's health prospects down the line, right? And they can look at protein markers in the blood, and they can accurately determine a person's health status, including what the current effects of abnormal genes or the reactions that the environment or the food or the toxins may have upon the genes of the individual. And we'll discuss that shortly. Now, that was prediction. Now, prevention is a new approach that we're seeing that, again, we're looking at the individual's genetic makeup, and it will help us determine whether people are at risk for certain diseases. And more importantly, even with epigenetics and genomics, the different genomic fields, the omics revolution, as it's called, how individual will respond to various treatments. And then we can provide guidance, customized therapeutic drugs, not only drugs, but also even medical foods, for example, and supplements. If we know what the genetic makeup of an individual is, and I have some examples that I'll give at the end. And then systems biology, technology is leading us to look at biomarkers and blood protein markers. And I mean, where we're moving is really just absolutely fascinating. This comes from the Institute of Functional Medicine, and I just got it from their website. And genetic, if you look at the top, it's right at the top, applies knowledge of genetic individuality to improve diagnostics and therapeutics, right? So personalized medicine is going to look at a combination of genetic vulnerabilities, environmental influences, try to reduce adverse drug effects or interactions, and it will be informed by evidence, right? But look at how complicated really everything else is, right? The multidisciplinary team that's going to be needed for the prediction, prevention, personalization, pharmacogenomics, nutrigenomics, the systems biology and the understanding that's underlying that, the laboratories, the pharmacological research, the nutrological research. So it's a complicated thing, and so as we move forward and knowledge starts to come out, I think it's yet to be determined where the registered nurse will end up playing a role. And in fact, we could create roles, we could find roles that we want to be in. If teaching is where you feel that you want to be, you could end up doing that. But there's a whole lot of other areas that are going to be created. In fact, they are being created right now as we talk. So I wanted to mention some trends that we're seeing for healthcare professional, right, as we move forward. And personalized medicine is really looking at individualizing healthcare, how to interpret data, medical history, family history, signs, symptoms, with the emerging omics technology. And the word omic is used, the omics revolution, because there's nutritional genomics or nutrigenomics, there's pharmacogenomics, there's proteomics, there's metabolomics, and so on and so forth. And I'll have an image here to give you a little bit of an idea. And this is going to give us a lot of power to tell the patients, you should not be eating this diet or that diet. And I'm sorry if any nurse ever comes up to you and says, oh, you shouldn't be talking about this genetic because, nurses don't need, I'm sorry. We do need to know, because you're a healthcare professional and you're going to deal with one of thousands of patients that you'll see in the hospital that had a heart attack, had a stroke, had an angiograph, had a stent placed, had a cabbage, had a coronary artery disease. And they're going to ask you, should I be eating fat, right? Is fat good or is fat bad for you? Well, it really depends on, do you have an allele, double allele, a type of allele? And I'll discuss this at the end of this lecture. So we do need to know these things. We're healthcare professionals, and it's our job to understand that we can't just say, well, good fat is good for you, bad fat is bad for you, because every individual is different. And this will be more true as we discover more alleles that affect how people process caffeine, how people process fats or process cholesterol. Is vitamin C good for you? Yeah, right. Vitamin C. Is vitamin C good for you? Linus Pauling, it's good for cancer. It's good for this. It's good for that. Well, we'll take a look at that as well, right? We'll take a look at that very shortly. But bottom line is that with this knowledge, medicine is really being revolutionized. So don't get stuck behind in the old world. Try to move your career and your understanding forward as you go through in your career as a registered nurse, as a healthcare professional. The transformation of the 21st century, we talked about this in the previous slide. You can pause it and go back over that. So, what's the potential of this future? So, nutritional genomics, yes, has the potential to change the dietary guidelines and personal recommendations. I have a feeling that the government guidelines on what we should or shouldn't eat eventually will be eliminated and will be determined on a person-to-person base. And nutritional genomics, also we call it nutrigenomics. It looks at the effects the nutrients have on the genome or the proteome or the metabolome, and that's why we call it genomics, proteomics, metabolomics. And nutrigenetics, as opposed to nutrigenomics, is the major goal of nutrigenetics is to try to find out what the effect of the genetic variation on the interaction between the disease and the diet is. So it's a little bit different approach between nutrigenomics and nutrigenetics. And so I got this image here just so you can get a picture of where we're going. So if you look on the right, all the way at the bottom, you have your DNA, and that's where genomics stands at, right? But as we move our ability to assess and look for biomarkers, ultimately it's all the way at the lower end, all the way at the top, metabolomics, where a lot of the promise seems to be waiting for us. And if you look at the left side, it's the same, but it's upside down. You have the genome and the DNA at the top, and all the way at the bottom, you have the metabolome. So when you think of metabolomics or the metabolome, the study of metabolomics is attracting a lot of researchers. A lot of money is going into this area because it's going to be telling us how sugars affect the individual, how nucleotides are going to, and look at the word that's all the way at the bottom, phenotype, function, how it's going to be changed. So if we can trigger, target, or even create and develop and utilize certain metabolites in our patients, we can change amino acids and lipids and nucleotides and sugars to affect the phenotype to prevent diseases or reverse perhaps even at some point. So it's amazing where we're going into. And so this is just to show you that at the DNA level, the genomics level, we're really looking at, there's 25,000 genes. Then you come down to the RNA with the translation and the transcription. There's 100,000 transcripts. Then okay, let's bring it down even a smaller level. Let's go down to the protein level. There's 1 million proteins out there, or even more. So proteomics, yes, it's vast. Yes, the power is there, but there's 1 million different things. With the biochemicals called metabolites, or the study of metabolites and how they affect us, we're really looking at a very small amount of compounds, which is why this is the area that a lot of research is targeting. And remember, the word epigenetics is altering the gene expression. You're not necessarily altering the gene, you're just altering the gene expression due to changing environment or lifestyle factors, right? They do not alter the DNA sequence, but rather how the DNA are going to be expressed. And epi means above, in genetics. So it's sort of a soft inheritance. So if you had your dad died of a heart attack, your grandfather died of a heart attack, your great-grandfather died of a heart attack, through this epigenetic approach, this soft inheritance, the food you put into your body through betaine, choline, B vitamins, proper nutrition, folic acid, methionine, the methyl groups can change ultimately the expression of your DNA and you can end up being a lot healthier, even if you have a lot of that history in your background. So keep that in mind. Epigenetics is really the new genetics. And we talked a little bit about what nutrigenomics was earlier, but it's basically looking at how nutrients, phytochemicals in plants, fruits, nuts, things of that nature contained in the food affect our genes. The interaction between food and gene. Remember, without fully changing the gene, but affecting the expression of it. So here in a nutshell, again, nutrigenomics focuses on the effect of the nutrients on the genome, the proteome, and the metabolome. Nutrigenetics focuses on the gene variants that are responsible for the differential responses to the nutrients. So a lot more of the research seems to be going down the track of nutrigenomics, although there still remains some looking at genetics, but they're very, very different. I'm not going to go into this in detail. I just brought this as an example. Polyphenol that green tea has can reduce the NFKB pathway that's associated with the breast cancer, right? So we don't need to know any of this, but we need to know that this already exists. So when you start to hear things such as green tea can be good because it inhibits phosphorylation of the HER2 or the EGFR, that receptor, ultimately helping breast cancer, well, then there you go. So there are a lot of natural compounds that can affect cancer cells through targeting the cellular signal pathways. And this is where the science and nutrigenomics is going. So guess who's investing in all of these medical foods and supplements? Well, most of the pharmaceuticals are, because they're seeing where the money is going and how much people are willing to pay for supplements and good food. These are just a few other examples of nutrigenomics and how they can affect cancer growth. I3C from a lot of your cruciferous vegetables. Curcumin, which is in turmeric, is not only anti-inflammatory and antioxidant, but it's a chemopreventive agent. Resveratrol, it contains phytolexin, which is present in grapes, mostly the skin, I believe. Mulberry, Persian mulberry, peanuts, they inhibit also the growth of various cancers. The lycopene, they say present in tomatoes. I think watermelon has more than even the tomatoes. And so you can see how a lot of this, as we move forward, is really going to change the nature of even how people treat diseases. Here I just created another chart. Again, people, these are just examples. I'm trying to get you to see a bigger, larger concept of where we're moving. Does diet affect our health? Well, we know that food has bioactive compounds on the left, right? So you can have, again, the C3G. You can have, and then in the middle you'll have where it comes from, right? So omega-3 fatty acids comes from fish oil, and not only is it a blood thinner, but it has some chemopreventive agents as well. So you get the point, is that diet clearly can affect our body. And tea, soybean, turmeric, as we talked about earlier, can have effect on the chromosome area that will really, when we go down to the metabolite level, look all the way at the bottom at what we're starting to discover, right? What do soybeans have? Genistein. What does watercress have, right? What does turmeric have? Curcumin. What's good about cabbage? Sulforaphane. I tell you, sulforaphane alone is showing tremendous effects on many conditions that people are manifesting and presenting with. Garlic, red grape, broccoli. Okay, so if people tell you that foods don't have power and can't make a change, tell them to go look into nutritional epigenetics and metabolomics. And you'll now know these terms, and you'll be able to. Do we know the details of this? Are we studying this in nursing school? No, but we are healthcare professionals, and we need to understand that. So this is just another slide. I'll let you look at it, how nutrition can go all the way down to change expression and even help people age slower, right? And slow oncogenes and decrease inflammation and many other things. So the sky is the limit as we go through the. So please keep informed in all of this information, because it'll benefit you, the people you meet. It'll benefit your children if you have them, your parents if you have them. It'll benefit yourself as well. I'm going to skip this right now. I'm going to try to go in. So I brought some examples of sulforaphane. I'm not going to go into that. I just mentioned how important it was. Antioxidants, we've had this talk in other lecture. They can be very important, but we have to watch out, because anything in excess can be good. So we want to balance out. The bottom line with antioxidants, which a lot of foods contain, is that free radicals ultimately damage cells, and they're like a toxin in the body, and having antioxidants that can prevent that damage to the cell is extremely important. But you'll only get that if you have a proper restored gut that has the right amount of pH in it, that is able to absorb and digest and absorb, I should say, into the body the proper nutrients that we need to eat to keep healthy. This was just from a research article that I found. I'm not going to go into that right now. There are a lot of problems with the nutritional obstacles that we're seeing in our nation right now are quite complex, right? There's diets that are high in meat and eggs, and meat and eggs can be good if you're getting them from the right source and eating them perhaps in small amounts. But sedentary lifestyle, fast foods, the quality of the food, the habit of not wanting to give up foods that we're accustomed to. If somebody's been used to eating steak, mashed potatoes, and gravy their whole life, try to tell them to start eating a diet with juice and take shots of some green weird vegetable, right? Wheatgrass. It's hard to change that. So not everybody can change that. We have to be very careful with just assuming, but we still need to inform our patients that there's a lot of, that they have options that they should help, that can help them, again, in moving forward with their healing process. Now, we always have to assess to see if they're ready and willing to heal, because I've had many patients who clearly, straightforward say, I have no intentions of quitting smoking. Absolutely. And I'll simply say, do you know what the risks are? Do you know what nicotine is doing? It's a vasoconstrictive. If you have diabetes and a history of stroke or heart attack in your family, and you're smoking, do you know, are you aware of this? And if they say, yes, I'm aware, then we have to respect the right of the individual to not want to quit eating fast food, quit eating ice cream, to not want to take up exercise. And so it's much more complicated than just saying, hey, change your lifestyle and we're good to go. Sometimes it's as simple. Again, I've repeated this in many other lectures. If you talk to a patient, if you can get them to change one thing, just one thing, if they drink soda pop that has high fructose corn syrup or aspartame, which is a toxin, it is a toxin, neurotoxin, and you can get them to quit that diet drink with that neurotoxin, and at one drink, just one thing, they never drink another diet soda in their life, or they never drink another high fructose corn syrup drink in their life. You've made a great advantage in success, as opposed to trying to tell them all at once that, well, now you need to go to a market in Napa and purchase food from the local sources and buy eggs from, not Walmart. And I mean, you could be overwhelming them. And at that point, they may not have the resources and the means to do so. But telling them what they can quit or eliminate from their diet is very doable for many, many patients. So again, all for one or none for all. So there is no fat is good for you or fat is bad for you. There is no coffee is good for you, coffee is bad for you. And when you guys start reading the literature, now with this lecture, especially the last few slides that I'm going to show you here, pay close attention, because now you'll understand this slide right here. Right? What's best for many people may not be good for you. What's best for some people or one individual may not be good for you. So is fat good or bad? Is coffee good or bad? Is vitamin C good or bad? Well, let's take a quick look at that shortly here. I'm going to skip this here. This is just sort of looking at the pharmacogenetic studies and how where we're at. We're on the left right now. We pool people together and we find a group of people that have adverse effects, a group that are poor responders, and a group that are good responders. Well, where we're going to is on the right, and this is where I'm getting to with coffee, fat, and vitamin C, and so on and so forth. In fact, so what I was going to say on the previous slide, when you start reading research articles, you will hear and read articles that say there was great statistical significance saying that butter was good for you. And others, there was great statistical significance that butter was really bad for the people. There was great statistical significance that coffee was good for you, or vice versa, that it was bad for you. And they could all be absolutely right. right? Where research has to start and is moving into is look at the right side of here. We're looking at people have the alleles, the AA genotype, or the AT, or the TT, and then we can match them with the drug, or we can match them and tell these people, for you, eating a low fat diet, you'll be safe. For you, you want to eat a moderate, you want to eat some more fat, you're okay. For you, you should stop drinking coffee. For you, you want to drink coffee, or it's okay, you're safer. Coffee is cardioprotective for you. So you can understand now how complicated a lot of the research that we have had done up till this day. A lot of it could be wrong because it was not looking at the epigenetic genotypes of the individual and the alleles. And I'll show you some examples not too long from now. So knowledge of your own genomic information can lead to better use of medication, absolutely, better management, and also what foods you can eat and utilize to prevent certain disorders and diseases. So let's get right in it. So I'm just going to talk about 3 examples, but I want you to keep in mind that I'm only presenting you 3 examples. You need to understand that every food, every liquid, everything you put into your body, whether it's a nut, whether it's a plant, whether it's a burger, whether it's from McDonald's, whether it's from In-N-Out, whether you cook it at home, anything and everything is going to have some type of effect on you. And we are, thank God, moving into a period of time where science is allowing us to now understand things in a way where before we were unable to. So I think that is a wonderful, magnificent step forward, almost like stepping onto the moon. In my opinion, even more grandiose than stepping on the moon, but that's just because I'm in the field of health and I'm interested in it. And I hope you guys do too, be interested in it, not just for that weekly or biweekly paycheck. So is vitamin C good for you, right? Linus Pauling, is it good for cancer? Is it, should we be consuming it? Should we be, how good is it? Well, we have to be careful. There's something called the G6PD deficiency. And we know that this deficiency can cause anemia. It can also cause hemolysis, there's drug-induced hemolysis. Even vitamin C could cause a certain amount of hemolysis. Now, that doesn't mean that if you have this deficiency, you can't get it. It just means you have to have extra caution. Right, and hemolysis is when the red blood cells, breaking open with certain foods or drugs are given, right? Like high doses of vitamin C, right? This deficiency is a hereditary X-linked recessive disorder, right? With an estimated worldwide prevalence of at least 329 million people. That means that 329 million people, unless they know that they're G6PD deficiency, if they went in to get a high dose vitamin C. But I will tell you, most labs, most places now that are giving nutraceuticals through IVs, they'll check you for your G6PD before you get a high dose vitamin C. That's just one example. And so we know this now, we're utilizing it. And now you know that if you go get your vitamin C ever in through an IV in mega doses, they probably will be checking your G6PD to see if you're deficiency, to work with you a little bit more cautiously than somebody who does not have this hereditary X-linked recessive disorder. Let's move into the second example. So what's ApoE4 gene, right? Well, it's a gene that we use it as a biomarker, right? And it's really an allele that's on chromosomes. Remember, we're talking chromosomes. And I believe the ApoE4 allele is on chromosome 19. You don't need to know that. It's a curious fact, though, that we do know, though, that if you have this ApoE4, there's E2, E3, and E4, especially those that are double allele E4s. It can put you at risk for a lot of problems, including cardiovascular conditions as well. And the ApoE4 is a gene that is involved in how we process lipids or how we process fats, right? And so we can use ApoE4 as a genetic risk profile to help us measure if you're at risk not only for Alzheimer's but for a lot of the cardiac conditions. Most people have ApoE3. About a quarter of us have a copy of the ApoE4, and these people are the ones that are really unlucky because they puts them at risk for cardiovascular disease and even for dementia as well. So yes, ApoE4 or possessing this allele is strongly correlated with Alzheimer's disease. And so I just wanted to throw in this word here. The ApoE4 heterozygotes have a 5-fold increased risk of not only Alzheimer's, but cardiovascular dementia problems in their lifetime. And remember, you guys need to know what heterozygotes and homozygotes mean. What does heterozygotes as opposed to homozygotes mean? If you have an ApoE4 heterozygote, you're people with 1 versus 2 alleles, all right? And if the people with the ApoE4 homozygotes, they're estimated to have staggering lifetime of up to 50 to 90% risk for the problem. So if they're homo, if they have 2 alleles, they're going to be a lot more at risk than the hetero. So this is, again, so is butter good for you? Is butter bad for you? Well, it really depends. Let me go back here. So the E2 carriers, if you ever do, 23andMe or one of these, it'll probably tell you what your ApoE is. And if you're an E2 carrier, you can clear fats fast, and you will need a high healthy fat diet and aerobic exercise. And it could be associated with either increased or decreased risk for atherosclerosis, but it depends on your lifestyle. So E2 really, you're not that much at risk because you can clear fats really quickly. E3 is the neutral. That's what most of us have, 70–80% of us. You're a normal metabolizer. You're not a fast, you're not a slow fat metabolizer, right? Carriers need a normal amount of healthy fat, normal amount of the good fat, obviously not the trans fat or the ultra saturated fat, and then we need to keep active, right? And then the one that we really need to focus on and remember, right, is the ApoE4. So when you're in that hospital and they ask you, hey, are fats good for you or fats bad for you? We now will be able to say, well, there's an apolipoprotein that we carry. Some of us are the E2, some are E3, some are E4, some are heterozygous, some are homozygous. And the E4 is the one that puts people, now there's only 14–15% of our population that has it. But if you have this allele, especially if you have 2 of them, you are going to be at risk for Alzheimer's disease, impaired cognitive function, reduced hippocampal volume, HIV, faster disease progression, and multiple sclerosis. So if you have a patient who has multiple sclerosis and they don't know anything about apolipoprotein E3, 2, 4, tell them about it, right? And they'll have unfavorable outcomes after TBI, traumatic brain injury, ischemic cerebrovascular disease, sleep apnea. They'll have accelerated telomere shortening, meaning they'll age quicker, right? So on and so forth. And it responds well to a low-fat diet, high vitamin D, and aerobic exercise. So if anybody ever tells you we don't need to know these things like apolipoprotein E, well, you might as well be saying that we don't need to be able to tell our patients whether fat is good for you or fat is bad for you. Because ultimately what it really depends on is if you tell a patient who has 2 E4 alleles to eat a whole bunch of the good fat, even the good fat, avocado and nuts. Oh yeah, eat a lot of avocados and eat a lot of nuts. They're very good for you. You're basically putting that patient at risk for Alzheimer's, for HIV, for multiple sclerosis, for cardiovascular problems, right? So you could be putting them at risk. So yes, we need to know these things. And as these things start to pop out and become known to us, we should become more and more familiar with it. And here I have 2 charts here. This sort of shows you the E2, the E3, and the E4. Let's look at the E4. The genotype, there's 3 and 4 and 4 and 4. And then they're at the highest risk for cardiovascular problem. The recommended diet for them is a very low fat diet. Now, somebody who has ApoE2, remember E3 is the normal. They need normal amount of fat. ApoE4 need low amount of fat. ApoE2 really they could eat, up to what we normally say the amount of fat to a 30-35% of fat is. So if you're one of those who eats a typical diet that consumes really low carbs and really high fat, and you have an apolipoprotein E4 or two alleles of that, you should really be eating a low fat diet, Now, the next slide is same thing, looks at the genotype, the metabolic expression, and what therapeutic interventions. So fish oil, for someone that has an E4, it'll lower the triglycerides, but not as much as maybe an E2 responder, right? A low-fat diet, compare the E2, which has one arrow going down, compare E3, has two going down, and E4 all the three arrows going down. That means that an E2 eats a low-fat diet and won't have that much effect on their LDL. But if an ApoE4 responder does eat a low-fat diet, it'll dramatically or drastically reduce their LDL. It'll even decrease their small, dense LDL. Now look at an E2. With a low-fat diet, it's going to put them at risk. It's going to increase the small, dense LDL. So they should, an E2 responder, a fast fat metabolizer, an E2 should not be eating a low-fat diet. An E4 should be eating a low-fat diet. So yes, we need to know that. I also like this slide down here at the bottom, because if you happen to know, you happen to pay the 80-90 bucks to get that, whatever that genetic testing is, if you happen to know that you're an E4 and you have cholesterol problems, well, we know that the simvastatin is going to be better, right, than somebody who's an E2 who is maybe an atorvastatin, pravastatin, or lovastatin is going to be better to treat their cholesterol. The E3, again, which is the majority of what our population is, no distinction. But if you happen to be an E2 or an E4, I think you would be really happy to be able to know, because then you really can target your own individual lifestyle. So when we say that certain diets are good, we need to be really careful with saying that low-carb, high-fat diets are good for everybody, because they're not. We know they're not. And as we learn more about this, then we not. Even the American Heart Association is now starting to come forward and say there needs to be an emphasis for focusing on gene influence on individual responses to dietary factors. And remember, as these large organizations start to come out with these, hey, we need to look at individual responses, it shouldn't be too long within our lifetime that we start to see that the government dietary recommendations are going to say, well, it depends, not just one pyramid, one triangle, or one circle saying everybody should eat nuts and everybody should eat this, and nope, that it just doesn't work like that anymore. And we need to know. And then the last one is about coffee, right? So is coffee good for you? Right? We may not need to remember what the allele is, right? What the name of the allele is, but we should be able to determine whether coffee is good for a person or bad for a person, and what makes it good or bad for a person. Well, it depends on if you are a slow or fast caffeine metabolizer. And it's as simple as doing a nutrigenomic biomarker check, which is quite common nowadays, for your CYP1A2 gene, right? A lot of people will like to tell you, oh, these complicated genetic words, we need to leave it for the specialists, we need to leave it for the doctors, we need to leave it for the... Well, if you want to leave these type of information for the doctors and for the specialists, then please don't be telling people caffeine is good or caffeine is bad. Just simply be honest and be straightforward and say, I don't know. But now with the information I'm going to give you, you'll be able to say that, well, a slow caffeine metabolizer, the alleles that they'll carry is the AC or the CC. And caffeine accumulation in these people may lead to high blood pressure and heart disease. Right? So if you said caffeine is good because you read an article that said caffeine is good to a slow caffeine metabolizer, again, we're in a genetics class right here, to a person who had the alleles AC and CC of the CYP1A2 gene, right? Then you would be putting them at risk for death. You would be doing them a disservice. On the other hand, if somebody is a fast caffeine metabolizer, meaning they have the alleles AA of the CYP1A2 gene, right? We know that they'll have a reduced risk of heart disease. So we really need to understand this, and that everything that we consume is probably in one way or another. I keep my eyes open at all times. And if you guys ever find out of one of these genes or alleles related to food or food intake, please bring it forth and show it to me, because I really thrive on knowing it. So bottom line with this whole lecture that we talked about, the DNA is not your destiny, because we now know we can change the expression of your DNA without changing your DNA through, thank God, epigenetics. Look at what this Time magazine from, I think it was 2019 or 18, was it 19, January. The new science of epigenetic reveals how the choices you make can change your can change your genes and those of your kids. So the interesting thing is that there might be a lot of people out here who will read articles like this and then talk to nurses, and a lot of your patients may have more information than you do. Be open to that. Learn from it. That's absolutely normal. There's just way too much information for all of us to be able to know, but that doesn't mean we shouldn't be informed. And we have this is the final slide here. So when we look at genetics, remember, genetics, ultimately the change of the sequencing of your DNA is ultimately really from a healthcare professional standpoint, and where we're moving in with our biomarkers and epigenetics is not as important as epigenetics. Epigenetics is really the new future of how can we influence the individual. Right? And again, remember that at the beginning of the slide, we talked about zip code affects more health than your genes. And that means that if you live in an environment that is industrial with high pollution in the environment, you're more likely to get sick. And that's because you're poor and you can't afford a better zip code. And here in the States, in our democracy, we create zip codes where people can go to better schools because they are in a better zip code. Well, don't think that health is not affected the same way. Zip code is more important than a genetic code. Now, if we tell the patients that are within certain zip codes that, look, it is because you're in this zip code, it is because you're exposed to drugs or cigarettes, or you're poor, or financially struggling, or your gut condition without probiotics, or you're taking antacids all the time, right? Or you have certain types of foods because that's all you have in your neighborhood, right? Well, telling them that informing them that these are external environmental influences that one can, to a certain degree, try to influence in one way or another. Sometimes a lot of these patients require case management, require social workers, physical therapists, occupational therapists, speech therapists. They require people to go do a visit in their home and look at what environment they're living in, right? If a patient is renting from a renter and has mold and that's causing asthma in their child, then we need to talk to the city to try to say, hey, look, the landlord needs to clean the mold behind there, or we need to get a way to support this family to move into a newer apartment or apartment that doesn't have mold, right? So the factors that influence health are tremendous. And yes, all of these environmental epigenetic factors will influence ultimately your patient. And that's it in a nutshell. So keep all of that in mind, and really start to really think in terms of individual. Every individual is very different, and you don't want to be telling a patient that is an E2 to eat a low-fat diet. That right? That fat is bad for them. You want to tell them to eat more fats, and you don't want to be telling somebody who has an E4 allele to eat nuts and high quantity of fats, because they're not going to be able to process it. It'll put them at risk as well. So you get the point, is we can't really be saying any longer with the information we're getting that one thing is good versus another, generically speaking, because ultimately it depends on the individual. And that's where we're at with genetics. That's where we're at with epigenetics. That's where we're at with the movement of omics, epigenomics, nutrigenomics. All right.